Welcome back to our study of population genetics. In this section, we're going to begin actually working out some population genetics problems. So as we go through these, I would certainly encourage you to pause the video and try to come up with the solutions on your own, and then hit play again and then check your answers. So the first thing we want to look at is we want to make sure that we're very clear on what we mean by a population. So to review, a population is going to be a group of interbreeding individuals. So this means that we have a bunch of males and females of the same species that are going to be producing offspring. And we're really assuming here that they're all breeding with each other. So we have random choice of mate and so on. Now, another important thing with a population is we have to assume that they are at least in close proximity to each other. So they have to be able to get near each other if they're going to be reproducing with each other. So if we had a species of fish in one lake, that would be a population. We could have the same species in a nearby lake, but it's very unlikely that fish in the two lakes would actually come into contact with each other and then they would be breeding with each other. So we would consider those two populations of the same species of fish to be independent populations. Now, here we have an example with cattle, and we're going to talk about coat color in cattle, and this is going to be a trait that's determined by a single gene. Now, this happens to be a case of incomplete dominance, and hopefully that sounds a little bit familiar to you from the last chapter, from chapter 14 during Bio 1. But if we look at these, this top one is what we would call a white cow. The middle one is what we would call red. And then the bottom one here is a color that we would call roan. And if we talk about the alleles involved in this and the genotypes that are involved in this, the white cow has two copies of an allele, which we're going to designate A with a little W superscript. So this one is homozygous for that particular allele. The red cow is going to be homozygous for a different allele, which we're going to call A with a capital R. When we have the heterozygous condition, that's the roan color that we have at the bottom. So the roan cow is going to be AW, AR. So those are our three genotypes, and since this is incomplete dominance here, the color of the cow is going to match the genotype. So it's very easy to match up the phenotypes with the genotypes. Now, if we start talking about the gene pool, if you'll recall from the last video, is basically the collection of alleles in a population. So we can call this the sum of alleles in a population. Okay, so if we have a population of these cattle, we would expect to have some white ones, some red ones, some roan ones. So let's throw some numbers in here. Let's suppose that we've got 150 of the white cows. Let's say that we have 40 of the red ones, and I'm going to say that we've got 40 of the roan ones as well. So if we want to count up how many alleles we have of the AR and of the AW, let's take a look at AW first. These cows right here, the white ones, they have two copies of AW. So we can add 150 plus 150. Okay, so that gives us a total of 300 AW alleles for the white cows. If we want to continue adding up AWs, if we come down here to the roan cow has one copy of AW and one copy of AR. So if we have 40 of these cows, then there is going to be 40 of the AW allele. So we've got another plus 40 to add on. So all in all, if we're talking about AW, we expect to have our 300 that we have from the white cows, and then we have another 40 to add on to that from the roan colored cows. So that's going to give us a total of 340. If we talk about the AR allele, the white cow does not have any. The red cows, again, we've got two copies of AR in those, so we would expect that we're going to get 40 plus another 40 because they each have two. So that's going to give us a total of 80 for the red ones. And then we can come down here to our roan cows and we expect to get another 40 because they have one copy of AR each. So for that particular allele, we have 80 plus our 40. So that's going to give us a total of 120. Now, if we want to talk about allele frequencies, I'm going to just clear the slide here. And if we put our numbers back up here, for the AW allele, we had 340. And then for the AR allele, we had 120. So we'll put that up here. So if we want to talk about allele frequencies, the allele frequency is just going to tell us how common is this allele in the population, or what is the proportion of the gene pool that actually has this allele. So the first thing we have to do for that is we have to add up the total of alleles that we have. So I'm just going to take my 340 plus my 120. So when I add those together, I'm going to get a total of 460 alleles. So it's 460 total alleles. Well, to figure out the allele frequency, we're really just taking the allele and we're putting it over, so the number of alleles over the total. So if we want to come up with the frequency for AW, we would take 340 and we're going to put that over 460. And then we're just going to divide the two. So go ahead and punch that into your calculator and see what you come up with. Okay, so I've punched it in, and the number that I'm coming up with here is going to be about 74%. Okay, so we're just going to round that to the nearest whole number in this case. And then if we do the same thing for our other allele, we're going to put the total, which is 120, and I'm going to put that over 460. And again, just punch that into your calculator and see what we come up with. And I'm coming up with 26%. Okay, so those numbers there would be what we refer to as our allele frequency. So the allele frequency is just telling us the percentage of the time that we would find that allele in the population. So the percentage of that allele in what we call the gene pool. All right, so the allele frequency, we would calculate that by putting the number of a specific allele over the total number of alleles. And we would convert that then into a percentage. If we look at yet another example, if we talk about some of the crosses that Mendel made, and maybe some things that are familiar to you from chapter 14 in Bio 1, we can talk about round versus wrinkled seeds. So if we represent this with an R gene, round is going to be 2 capital R's, and then or, this case, and wrinkled would be 2 lowercase r's. So let's assume that we've got these individuals, and we know what their genotypes are, and we've got a population of them. Let's just throw some numbers out for each of these. Let's say that we have 14 of big R. Let's say that we've got 52 of the heterozygous, and let's say 4 of little r. And we want to figure out what the frequency of each of these alleles is. Well, if we go through the steps that we did before, for capital R, I put it right here, capital R, we're going to have from these guys right here, remember that they're homozygous, so they're going to have 2 copies of capital R each. So we have here basically 14 plus 14, and that's going to give us a total of 28. And we can also expect to get 1 copy from each of these individuals, so that's going to give us 52. So for R, I'm going to take 28 plus 52, and that's going to give me my number of R's, capital R's. And then if we look at the lowercase r, we've got 1 copy each from the heterozygous one, so that's 52. And then my homozygous little r is going to give me 2 copies each. So I've got 4 plus 4, or in other words, I've got 8 from those. Now, if we just take those numbers that we have of each specific allele and we divide it by the total number of alleles that we have, we can come up with the allele frequencies. So let's do that. So for all of them, if we add them together, keep in mind that We're getting 28 total alleles from these guys. We're getting 52 plus 52 from these, and we're getting 4 plus 4, or 8, from this one. So let's add those up. So I've got 28 plus 52 plus 52 again plus 8. I'm coming up with a total of 140 alleles. 140. That's my total number of alleles, so I'm going to put that on the bottom right here and here. And so now we're just converting it into a percentage. So for capital R, go ahead and plug that into your calculator. Okay, and what I've come up with for capital R is going to be 57%. And then for lowercase r, we've got 40, I'll round it to 43%. Okay, and notice in this example, as well as the last example, when we considered the different alleles that we had in the population and we account for all of them, they do add up to 100%. Okay, it does in this example, it also does in the cattle example that we just went through. So that's how we would calculate allele frequency. Okay, so if we have a population that is not evolving, then we expect the allele frequencies to remain the same from one generation to the next. So if we look at one generation and we see 57% capital R and we see 43% lowercase r, if we looked at that same population 3, 4, 10 generations later or 100 generations later, we would expect those frequencies to be exactly the same. Now, if we have evolution actually happening, so if the population is evolving, we're going to see those numbers change. Okay, so as we said, when a population is not evolving, the frequency of the alleles and also it's going to be the genotypes in a population's gene pool will remain constant from generation to generation. Now, this is what we call genetic equilibrium, when everything stays the same from generation to generation. Two individuals independently studied this, and they came up with a theorem which we call the Hardy-Weinberg theorem. And so this was put out in 1908, again, independently by these two individuals, and this is really the foundation of what we call population genetics. So what they were saying is that if the population is not evolving, everything stays the same. So we want to think of the alleles in the population, the gene pool, as just being a pile or a collection of alleles. Okay, so what we're seeing inside here, inside this box, is what we want to call the gene pool. Okay, so this is my gene pool. We don't want to think about the individuals in the population anymore. We're just talking about all of their alleles added together. So notice in this picture, we've got some blue or purple dots or alleles, and we've got some green ones. So we can calculate an allele frequency for these different dots. So if we talk about the blue ones, keep in mind that if we're calculating allele frequency, we have to add all of these up. So for the blue ones, if you want to go through and add them up, I've already added them up. We have 20 of those. And we have a total number here of 32. And so my frequency for my blue alleles is going to be 62.5%. And then I can also calculate it for the green ones. And the green ones, I have 12. And I'm also going to put that over the total here, which is 32. You can divide them out to come up with the frequency. And in this case, it's going to be 37.5. Okay, so those are my allele frequencies. And this is my gene pool. And this is specifically what we're talking about when we talk about Hardy-Weinberg. So if we go back to our cattle, again, we had a certain number of the AW allele, and we had a certain number of this allele, and we no longer want to worry whatsoever about what each individual cow has. So we don't really care about whether they are heterozygous and they're going to have one or each, and the heterozygous ones might want to mate with the heterozygous ones. We're not worried about any of that. We're just worried about the total number that we have of each one of those alleles. So we had 340 of this one and we had 120 of this one. That's our gene pool that we're going to be working with. Let's look at a problem here. So in this case, we've got some carnations or snapdragons. It's a similar example, regardless of which one of those two flowers you're looking at. And we want to calculate some allele frequencies. So we've got a certain number of plants. We've got 320 plants that have red flowers. We've got 160 with pink flowers, and then we've got 20 with white. Again, to calculate allele frequency, all we're doing is we're taking the number of the allele and we're going to put that over the total. And it's as simple as that. It's important that we keep in mind that each individual is going to have two alleles, and that is going to be because they are diploid individuals. So if we're looking at our homozygous red ones, so these are the red ones. I know it's hard to tell in this picture, but this is red, this is pink, and these would be our white ones. So as we look at these, keep in mind that the red ones are going to have two copies of the capital R or CR allele, so that gives us the 640 that you see right here. And then the heterozygous ones, or the pink ones, also have one copy each. So if we have 160 of those, we have 160 of that particular allele, and we can add those together. When we add those together, we get the 800 number that you see there, and we're dividing that by the total number of alleles, which, if this is a population of 500 total, so 500 individuals, if you add across here, every individual is diploid, so we have a total of 1,000 alleles. That gives us 0.8 or 80% for the CR. So CR is going to be 80% based on these numbers. If you go through the steps with the CR or the CW or the little r allele, we're going to get 20% or 0.2. So that's how we calculate allele frequencies. Make sure that you are able to calculate allele frequencies if you're given a population of individuals of different phenotypes. So if we go to what the Hardy-Weinberg actually states, for one of the alleles, we're going to call them P sub A or PA, and then we have one that we're going to call Q. Okay, so P and Q is what we kind of reduce this down to. We tend to use the P to represent the dominant or the capital letter, and we tend to use the Q to represent the lowercase or recessive letter, but it really doesn't matter which one you use. Now, what they said, Hardy and Weinberg, is they both said that if you add together all of the frequencies of the allele of a given gene, it should add up to 1. So basically, P plus Q, the frequency, is going to add up to 1. And we did see that happen in the last couple examples that we looked at. Now, they took this a step further. So they said if P plus Q equals 1, then if you square that, it should also equal 1. So if you remember your algebra, if we expand this out, multiply it out, this is the equation that we come up with. This is the Hardy-Weinberg theorem. This is an equation right here that you want to know. So what this equation tells us is it's basically what we can use to come up to take the allele frequencies and use that to come up with the predicted genotypes that we would have in the population. Make sure that we understand the difference between what we're calling an individual allele and what we're calling a genotype. A genotype would be the genetic makeup of an individual in the population. So that would be the homozygous dominant or the heterozygous ones. So if we use this particular equation and we have allele frequencies, then we should be able to predict the percentage of the different genotypes in the population in each generation that we go through. Since P represents the capital letter generally, if we take P and we square it, then we're going to get the frequency of the homozygous dominant or the big A genotype. Since Q represents the lowercase a, if you square that, you get the genotype of little a. And then in the middle here, the 2pq, that's going to give us the frequency of the heterozygous individuals. Now, the reason for the 2pq is if you remember the Punnett square, if we make this cross, keep in mind that there were two ways to get heterozygous. So we've got this one, plus we've got this one over here. So that's really where the 2 comes from, the fact that we can make those heterozygous individuals in two different ways.